Good morning, everyone. Firstly, thank you to MMF for inviting me to talk to you today. So my name's Tim Gilbert. I'm one of the surgical registrars up in Liverpool, and I'm currently undertaking a PhD at the University of Liverpool, looking at developing a patient-derived model of hilar cholangiocarcinoma using precision-cut tumour slices. This is actually something you're going to hear a little bit about more from my colleague, Dr. Laura Randall, presenting that afterwards. I'm actually standing in for Mark today, who we had hoped would be here presenting you an update on some of the single-cell sequencing work that's been conducted in Liverpool, which has been MMF funded. But as Mr. Mallett's touched on, unfortunately, he's not able to join us today. But we still felt it would be important for us to showcase what MMF-funded research has been conducted in Liverpool. And I'm going to be representing some of our earlier sequencing data that Mark produced, looking at the identification of actionable targets in peri-hilar cholangiocarcinoma. So just in terms of what we're going to discuss today, so firstly a little bit of background as to why it is that we did the study that we did, what the aims of that study were, how we went about setting about to achieve those aims, going to discuss some of the results of our study, and just touch upon some areas of further study that we're going to be conducting. So first of all, a little bit of background. As we've heard a lot about during this conference, genetic sequencing is playing an increasingly important role in how we're understanding and treating cancer through the identification of actionable drug targets. Historically, sequencing studies in cholangiocarcinoma were limited compared to sort of the other related GI cancer types, but over the last 5 to 10 years, there's been an explosion in the number of sequencing studies conducted. However, when we looked a little bit deeper into the literature that was being published, we saw that the vast majority of these sequencing studies were conducted in patients with intrahepatic cholangiocarcinoma, despite this being the least prevalent subtype. And this is important as we know that the three various subtypes of cholangiocarcinoma behave in very different ways. Now, of course, extrahepatic disease has been sequenced, but again, when we looked at the underlying published literature, a large proportion of these studies have been conducted in the Far East and Eastern populations, where we know that they have a different underlying etiology to that we see within the West. So chronic hepatitis infections and fluke disease. So that led us to ask the question is, what are the relevance of these published studies to a UK or Western cohort that primarily see idiopathic peri-hilar disease? And this is important as we know that sequencing studies can translate into efficacious treatment targets, as we've seen in intrahepatic cholangiocarcinoma with IDH1 and 2 mutations and FGFR2 fusions. And it's these sort of actionable drug targets that are lacking within this peri-hilar cholangiocarcinoma cohort. So the aims of our study were to characterize the mutational landscape in a resected UK peri-hilar cholangiocarcinoma cohort, validate the presence or absence of what are already the published mutation in publicly available data sets, see if we could elicit any actionable drug mutations from our data set, and if we can identify any potentially novel or new mutations to the hilar cholangiocarcinoma scene. And putting all that data together, whether we can infer any new or characterize the underlying biology of cholangiocarcinoma further. So in terms of the technology that we employed, it was primarily whole exome sequencing in combination with some targeted sequencing studies. And these are both next-generation sequencing techniques that look at DNA within the protein coding regions of exons. We know that's important because around about 85% of all disease-causing mutations are within these regions. In terms of the tissue samples that we had available, so at the onset of the study, we had access to 45 full and fixed resected specimens, and we supplemented that with a fresh tissue acquisition protocol, which we gained an extra 20 fresh frozen cases. In both cases, we had matched tumour and normal duct. All these specimens underwent consultant histopathology review and underwent microdissection to boost the tumour cellularity to ensure that what we were sequencing was mainly tumour cells in the main. These samples all then underwent quality control, and we took 25 samples forward to whole exome sequencing and a further 20 through to targeted sequencing, and they matched up with the fresh frozen samples that we managed to obtain. The research was conducted within the Liverpool Centre for Genomic Research, and the whole exome sequencing study was conducted on the Lumina HiSeq 4000 and the targeted series on the ION PGM sequencing 200 kit. We achieved a depth of greater than 50x on the exome series and greater than 1000x on the targeted series. In terms of the bioinformatics, once we performed our sequencing, we then aligned this to the human genome and we performed a somatic variant analysis. We considered variant allele frequency of greater than 1% to be significant, and anything between 1% and significantly less than 50% to be a somatic mutation, anything approaching 50% or over to be considered germline. We then screened these outputs against publicly held databases of cholangiocarcinoma patients within DB-SNP and COSMIC. And we also then screened our sequencing data against the OncoKB database of actionable drug targets. And then finally, we performed a Geneset enrichment analysis. So in terms of our results, so just some sequencing statistics initially. So we achieved on-target mapping within the exome series of 76%, and on-target mapping in the targeting series of 99 to 100%. When we merged these two data sets together, we had a median number of somatic mutations per megabase of 13.57 per megabase pair, with actually a fairly significant range with some outliers at the top end. This all correlated with a moderate tumor mutational burden, and it's something that is actually higher than what we've seen previously published in intrahepatic cholangiocarcinoma and other sort of related hepatopancreatic cancer types, pancreatic cancer. And we know this is important because tumor mutational burden correlates with your neoantigenicity, and that in itself correlates with immune cell infiltration and ultimately potentially improved response to immunotherapy. Moving on to the mutational landscape itself, so when we compared our results to the publicly available cholangiocarcinoma sequencing data sets held within COSMIC, we saw some quite stark differences. So on the left-hand side, we can see the data that's held within COSMIC and that we obtained in Liverpool. So these are the top 6 most frequently mutated genes held within COSMIC. As you can see on the left, 26.6% P53 mutation rate, and in Liverpool, that was up to 50%. KRAS was roughly equivalent, but as we move down the list, we start to see some significant changes. So IDH1 and 2 and BAP1, 10 and 8% in COSMIC. Mutations may be more associated with intrahepatic cholangiocarcinoma. They were absent at the 1% variant allele frequency within our cohort. What was also interesting is then when we looked at some of our high frequency mutations within our cohort, we saw high frequency mutations both in mTOR, ABL1, NOTCH1, NF1, and EGFR. And again, when we compared that to the COSMIC dataset, there were some significant differences. Now, each of these are all established cancer mutations in themselves, but as we can see here, not ones that are particularly associated with cholangiocarcinoma, and certainly not on the frequencies that we saw within our cohort. And this was interesting because when we took through these high-frequency mutations and then screened them through the OncoKB database of actionable mutations, the vast majority of these mutations that we see at high frequency are potentially actionable or have actionable drugs in other cancer types. And overall, when we looked at the total number of patients that had a potentially actionable mutation within our data set, that was approaching 50%, near enough 50%. Just to summarize those results there, so we've seen that we've got a moderate tumor mutational burden within our cohort. We appear to have a distinct genomic landscape within our hilar cholangiocarcinoma cohort with some high prevalence mutations in the genes as described. And importantly, up to 50% of our patients had at least one potentially actionable mutation. And that obviously has implications going forward, as we've heard throughout this conference, about looking at personalized therapy and how we can get more of our patients involved in sort of contemporary basket trials. As Mr. Malik touched on, so part of the benefit of doing whole exome sequencing within our study was that it gave us the potential to identify potentially novel mutations, and this is one that we found within our data set within the MAP3K9 gene, and this is something that appears to be novel to certainly the hilar cholangiocarcinoma setting. So we had a CCT deletion in 22 out of 25 of our patients, so 88%, and this was present at a somatic variant allele frequency in 10 tumours, and then a germline frequency in an additional 12. Now, as yet, we're unclear as to whether or not this mutation is leading to gain or loss of function within this pathway. But looking at the published literature, we can see that inhibition of the MAP3K9 pathway in models of pancreatic and esophageal cancer does reduce cancer cell proliferation. However, on the flip side of that, we have actually seen as well, again in the published literature, that loss of MAP3K9 function is associated with chemoresistance, and that's been described in melanoma. So this is work that we're looking to take further in Liverpool. Clearly, this is an area of interest and something that we want to explore further, because MAP3K9 is a potentially targetable pathway. Finally, just to touch on the Geneset enrichment analysis that we performed. So having got all of our sequencing data together, we performed this final analysis looking at enriched cellular pathways, and we found that we had 21 cancer-related pathways that were significantly enriched at the 5% false discovery rate. The most significantly enriched of these pathways were pathways that pertain to host immune response, with Dectin-2 being the most enriched innate response pathway, with PD-1, CD3 phosphorylation, and ZAP70 translocation being the most enriched adaptive pathways. And these are all pathways involved in T-cell receptor activation. So putting all this sort of information together suggests that the immune response has an important role to play in hilar-cholangiocarcinoma progression. And this is something that we're going to be looking at further within our precision-cut tissue slice model as we look to characterize further the immune profile within our specimens and whether or not that can be modulated. So in summary, we appear to have a distinct mutational profile within our Liverpool hilar-cholangiocarcinoma cohort, certainly compared to that that's available within the publicly available data sets. We appear to have a novel mutation within MAP3K9, which is high prevalence, both at somatic and germline mutations. And this requires further work from ourselves to establish the significance of this. Within Geneset enrichment analysis, we have multiple enriched immune response genes, and this is something that we're looking to explore further, as I've said, through our precision-cut tissue slices and related studies. Thank you from me. Thank you for listening. Obviously, a massive thank you to MMF. None of this research is possible without your support. Obviously, thank you to Mark Quinn, who's standing in for today, who's generated all the data for this study and presenting on his behalf, and to the wider team that's enabled the success of this project. Thank you.